Ladies and gentlemen, welcome to the lecture Vaccine Components and Immunization Techniques, the Principles of Immunization Techniques. Before I actually start my lecture, it is very important to define what a vaccine actually is. Vaccines belong to the class of biological medicinal products, meaning that these are complex products and also meaning that the active substance, which is the vaccine antigen, is derived from a living organism and cannot be chemically synthesized like the small drug molecules like aspirin and ibuprofen and many others can actually. There are a number of biological starting materials which are needed in order to produce vaccines, like cells of human and animal origin for the propagation of vaccine viruses, like embryonated eggs used for the same purpose. There are virus strains and viral vectors, which are living organisms as well, just as are bacterial strains or recombinant yeast vectors, which are used to produce recombinant antigen vaccines. Slide number 3 then gives you an impression what an ideal vaccine should actually do. First of all, it should mount a specific immune response against a circulating pathogen, which on an individual but also on a population basis should meet a number of objectives. First of all, it should protect from infection. That's the ideal case if this can be achieved by a vaccine. But it would also be sufficient just to protect from disease, allowing infection but preventing disease. There are many vaccines which do not fully comply with these ideal objectives. There are many vaccines, at least in certain age and risk categories, which are just able to reduce morbidity and mortality, which is okay as well. There are vaccines which, in addition to the previous bullet points, may lower transmission rates of pathogens from a protected, meaning vaccinated individual, to a non-vaccinated neighbor individual. And if this effect is broad enough, we talk about herd immunity, which means that a fully vaccinated society can actually protect those who don't want or who can't be vaccinated. And this effect is a very valuable one because it helps to eliminate infectious diseases, and in the ideal case, it helps to eradicate pathogens from the globe. But this final point has only been achieved once, about 3 decades ago, when the variola virus was actually eradicated. Variola virus causes smallpox, and that's a well-known success achieved by successful vaccination programs. Slide number 4 explains a bit how vaccine efficacy is actually achieved. Which factors, which elements are actually needed to produce a functional and efficacious vaccine? First of all, it's the choice of the right antigen. Not all pathogens or not all parts of a pathogen, a bacterial or a viral vector, are actually suitable to produce antigens which do mount an effective, meaning neutralizing or protective immune response. So it's very important to know about antigen function and choice of antigens. If this has been done, the dose is also a crucial thing, meaning to define how much of this antigen do I actually need in order to swiftly and efficiently mount an immune response of high quality. And even that is not enough. It's also important to determine the right number of doses needed to be injected or given in order to achieve the maximum vaccination effect. So many vaccines for that reason are actually given in a 2-dose, in a 3-dose, or even in a 4-dose regimen in order to achieve a robust and long-lasting immune response. And in that context, the vaccination regimen is also crucial. That means to determine the time window in between which the individual doses need to be given in order to be fast on the one hand regarding establishment of a functional immune response, but on the other hand, also to take the time in order to get a long-lasting and efficient immune response, preventing that a certain vaccine needs to be given regularly. That means every year or every 2 years. Then the formulation, meaning the solution which is in a syringe or in a vial, is also crucial because there are many ingredients needed in order to make vaccine antigens stable and to transform them in a form which actually induces maximum immunity, like for example by adsorption to aluminum hydroxide or by adding other suitable adjuvanting systems, things I'm going to talk about in a minute. And then the method of administration is also crucial. You know, there are very few vaccines which can be given nasally. It's actually only a single influenza vaccine which is licensed for that purpose. Most commonly, the intramuscular or the subcutaneous route is actually used, but there are also vaccines that work through oral administration. All things we need to deal with in a couple of minutes. Slide number 5 adds some additional considerations which are important when vaccines are developed and filed for licensing. It's the primary immunization which is crucial. And primary immunization means what needs to be done when a vaccine is given the very first time to a previously unimmunized or unvaccinated individual. So the two things I talked about already are important to know about how can an immediate protection be achieved as fast as possible and what is needed in order to provide a long-term protection through vaccination. And then, in contrast to the primary vaccination, a booster immunization is also important to consider, meaning to investigate how long after primary immunization does my immune response last before I need to be revaccinated in order to refresh an optimal immune status. So there are two questions which are important. First of all, is there a booster immunization needed after a while? And if it is needed, when is the appropriate time point? And many vaccines which require a booster immunization will require that after 10, 15, maybe 20 years. And other vaccines after primary immunization need not to be refreshed, meaning that they actually induce a lifelong protective efficacy. Besides efficacy and effectiveness, vaccine safety is another important parameter because vaccines are given to a healthy population, and that means that the number and severity of side effects should be reduced to an unavoidable and low level. And both together, immunogenicity, efficacy, effectiveness, and vaccine safety provide you with a benefit and risk calculation. And for a vaccine, the benefit risk ratio should always be in a way that the benefits outweigh by far the unavoidable risks of vaccination. Slide number 6 explain the target populations. So that means all of us are confronted with vaccines from our earliest day onwards. So there are preterm babies which are exposed to certain vaccines in order to avoid life-threatening disease patterns like rotavirus infections or RSV, that's an acronym for respiratory syncytial virus, which is not given as a preventive vaccine but as a monoclonal antibody formulation in order to prevent this really, really threatening disease for preterm babies. Most of the vaccines are then given a bit later to infants and toddlers in their first and second year of life. It's a broad range of options, which is given here, most notably the DTPA-based vaccines, which is diphtheria, tetanus, and acellular pertussis components, which are combined with IPV, which is inactivated poliomyelitis vaccine, Haemophilus influenza type B vaccine, and hepatitis B vaccine. That all comes in the same syringe, but beyond that, there are other vaccines like meningococcal vaccines, A, B, C, Y, and W, pneumococcal vaccines, the MMRV, measles, mumps, rubella, and varicella vaccines, and rotavirus vaccines. Many of these vaccines are available as combination products, as to be explained a bit later. Older children and adolescents have specific vaccination groups as well, in particular vaccines against sexually transmitted diseases, like, for example, hepatitis B and the human papilloma virus vaccines. Adults are, in principle, the key target group for many travelers vaccines like rabies, yellow fever, Japanese encephalitis, hepatitis A, typhus, cholera, and there is a growing number of additional vaccines coming up, like, for example, malaria and dengue virus vaccines. Slide number 7 continues with the target population. They are, of course, the elderly, where specific vaccination recommendations exist, for example, against influenza and pneumococcal diseases. Other risk groups are pregnant women, and they are really in the focus right now, and specific vaccines are already recommended to be given to this risk group, like influenza vaccines, pertussis vaccines, tetanus vaccines, and this is under investigation. There's no such product yet available. Also RSV, again, that's a vaccine against respiratory syncytial virus infection. And all these vaccines do not only protect the mother, but also the baby when it is born until it is old enough to get one of the vaccines mentioned previously for childhood immunization. There are patients that need specific vaccines, for example, before or during a hospital stay, and there are frail individuals which should be vaccinated in order to prevent that their weak health status is further weakened through acquiring an infectious disease which is vaccine preventable. Slide number 8 lists a number of expectations the average population has actually towards vaccines. So everybody is expecting that these products should be highly efficient. They should work immediately, which is hardly achievable, but this is one of the expectations. There should be easy, fast, and painless administration, which is the fact in most cases, because the needles used nowadays are almost painless, and there are also vaccines which can be given orally and nasally, as I already explained. And there should be no side effect. But this is also something that cannot be fully achieved. Vaccines are safe. There are no side effects, or only a few side effects observed normally, but there is no vaccine without a side effect. Slide number 9 takes us to the different vaccine classes that exist, and the most prominent ones are certainly the life attenuated vaccines. We know them, or most of them, from our own childhood vaccination programs. Measles, mumps, rubella, varicella vaccines are commonly given to a European population, like rotavirus vaccines. The other ones, yellow fever, BCG stands for Bacillus calmette-guerin, which is a vaccine which is not given anymore in many countries, and cholera and typhus vaccines are only given as travelers vaccines. There are live reassortant vaccines, which are different from the live attenuated vaccines because this is a different way of constructing them. They are genetically modified organisms because most commonly they have an animal part plus a human part, meaning there is an animal virus which works as an attenuated virus in the human host, and the specific immune response is mounted by adding the relevant components from the human viruses which infect humans, causing disease there. So these are reassortants, and there is a rotavirus vaccine, which is the most prominent example in that way. And these live reassortant vaccines work perfectly well, and they are very, very safe. Another interesting group are the live viral vectored vaccines. These are genetically modified organisms as well. Dengue vaccines are the first one which are licensed in endemic countries like Brazil, the Philippines, and Mexico. And here you take a viral vector backbone like an adenovirus vector or a yellow fever vector or a VZV vector, which stands for vesicular stomatitis virus. And the relevant antigen of a pathogen, which actually mounts a specific immune response, or the genetic material coding for that is inserted into that vector, which makes a recombinant vector, and expressed in the body after these vectors have been injected or given by another route of administration. And that mounts a very effective and specific immune response against the recombinant antigen. A similar concept would be feasible for bacterial strains, bacterial vectors, but no such vaccine exists actually. Slide number 10 continues with the different vaccine classes. The majority of vaccines comes as inactivated vaccines, meaning that either the whole virion or parts of a vaccine virus or a bacterial vaccine strain are inactivated by certain chemical agents like formaldehyde or propionic lactone, making it very safe and tolerable. And the most known examples for whole virion inactivated vaccines are hepatitis A vaccines, influenza vaccines, specifically those used in a pandemic context, Japanese encephalitis, as well as poliomyelitis vaccines. For the whole cell bacterial vaccines, pertussis vaccines used in the past but still used in many countries are an example for an activated whole cell based vaccine. Since many whole virion and whole cell based vaccines turned out to be rather reactogenic, specifically when they were given repeatedly, the concept of purifying most relevant parts of vaccine viruses or bacterial strains was further developed, and this results in so-called split vaccines, where a whole influenza virion is actually split by using or adding mild detergents, and or by further purifying these split antigens, formulating them into subunit antigens, where only very specific and well characterized components of a vaccine virus or a whole cell microorganism are actually used in the final formulation. And these are influenza vaccines and pertussis vaccines as well-known examples. Slide number 11 continues with vaccine classes. They are so-called component vaccines, where actually many well-characterized proteins of a certain vaccine strain are extracted and purified So there are acellular pertussis vaccines containing up to 5 or 6 components from the original Bordetella pertussis bacterium. There are polysaccharide vaccines. There's no longer a protein which is used as a vaccine antigen, but only the outer polysaccharide surface or parts of it, which are present on the surface of many, many bacterial strains. They come as conjugated vaccines as well. This is something which we will talk about a bit later. But many, many bacterial vaccines like Haemophilus influenza type B, meningococcal and pneumococcal vaccines do not contain a protein antigen anymore, but a polysaccharide chain which is purified from the original pathogens and used as a vaccine antigen. Another technique is to purify the toxins from certain bacterial strains, like from pertussis strains, diphtheria strains, or tetanus strains, and inactivate them again by using formaldehyde or other inactivating agents. And then you transfer a toxin which can be lethal to a toxoid which has lost all its pathogenicity but has remained its immunogenicity, and simply these toxoids are sufficient in many cases as a single antigen to induce protective immunity in a vaccinated individual. Slide number 12 finally concludes on the vaccine classes. As I said before, there are recombinant vaccines as well, like hepatitis B and the human papillomavirus vaccines. In that case, the antigen coding for important proteins which induce a neutralizing immune response are cloned in an expression vector. In this case, it is yeast, Saccharomyces. This is not itself injected into patients, this recombinant construct, but used for production in order to produce recombinant virus-like particles, which are then purified and used as a vaccine antigen. And finally, there are some investigational concepts like nucleic acid-based vaccines, just using either a recombinant plasmid DNA or there are also RNA vaccines coding for specific vaccine antigens which are expressed in the human cell following immunization. But these are concepts which only exist in a preclinical investigation program or in very, very early phase 1 clinical trial studies. Besides the vaccine classes, and I'm now turning to slide number 13, there are a number of product particulars which also define vaccines or vaccine classes. The most common one are the so-called adsorbed or adjuvanted vaccines, which applies for most inactivated vaccines. And this means that the vaccine antigen is actually adsorbed to aluminium hydroxide. This does not come as a salt, and the formula is depicted on the right. It's a very complicated three-dimensional matrix consisting of aluminium hydroxide or aluminium phosphate, which is used to adsorb the protein-based vaccine antigen, making it more stable but also more immunogenic. And there are further adjuvanting systems which are called novel adjuvanting systems, which help a lot to increase the immunogenicity, specifically of highly purified subunit antigens, by adding substances like MPL, which is monophospholipid, or so-called oil in water-based adjuvanting systems, or a combination of these in order to significantly enhance the immunogenicity of otherwise, barely immunogenic vaccine antigens. And in my second presentation, later on I will focus on these aspects. But these are important product particulars, making vaccines really efficient, meaning that potent adjuvanting systems are added to protein or also polysaccharide-based vaccine antigens. Slide number 14 gives you some more examples on vaccine classes, on product particulars. There are the previously mentioned conjugated polysaccharide vaccines, and that means that the sugar structure doesn't come as an antigen by itself, but is conjugated to a protein carrier, which very often is also a bacterial protein, in order to specifically and significantly enhance the immune response against the polysaccharide structure, ensuring fast induction of protective efficacy, but also persistence of immune protection over time. So these are very, very important aspects and developmental steps in the evolution of vaccines. Virus-like particles were also and already briefly mentioned. Hepatitis B, human papillomavirus vaccines, influenza vaccines, and malaria, or the only existing malaria vaccine, are relevant examples. And that concept actually alludes to the fact that structures which are very similar or very related to the original pathogen should also be as immunogenic as the original pathogen. And that's the reason why vaccine developers often try to mimic the structure of the original pathogen by producing virus-like particles. And that's a very effective way of producing highly efficient vaccines. Slide number 15 then gives you even more product particulars. There are multivalent vaccines against the same disease, but also against different diseases. Against the same disease means that very often pathogens circulate as certain types, serotypes, and that would mean that a vaccine would be needed against each individual serotype. So in order to address that level of complexity, many vaccines with different strains circulating come as combination vaccines, as multivalent vaccines like polio vaccines, which incorporate 3 different strains or types, 1, 2, and 3. Meningococcal vaccines, where different serotypes are combined in a single syringe. Pneumococcal vaccines, where up to 23 serotypes are assembled in the same vaccine. There are also combination vaccines against different diseases. The previously mentioned DTPA vaccines that may come together with IPV, HIB, and Hep B components. These are the so-called hexavalent vaccines given in early infancy. The measles, mumps, rubella, varicella vaccines, but also many travelers vaccines like hepatitis A and hepatitis B, or hepatitis A and typhus, come as combination vaccines. And if you look at the vaccination schedules or the number and kind of officially recommended vaccines, it's easy to understand that addition of new vaccines into existing vaccination schedule can only be realized when they are part of a combination vaccine. If all these vaccines would come as monovalent vaccines, realization and implementation of vaccination regimens, officially recommended vaccines, would become quite difficult for both, for an individual, for mothers and their child, but also to general practitioners. So combination vaccines, multivalent vaccines are very important things to consider. And further to that, concomitant use is another strategy to simplify vaccination schedules, meaning that combination vaccines could be given at the same time but in a different arm, like the hexavalent DTPA vaccines that can be given together with measles, mumps, rubella, and varicella vaccines, or with pneumococcal vaccines, or the MMRV vaccines can be given together with pneumococcal vaccines, and so on and so on. The important thing is that this needs to be clinically investigated in order to learn about the safety of this concomitant use strategy, but also in order to learn that this second vaccine given together with the first one does not result in a reduction of immunogenicity, efficacy, and effectiveness of either of these components given, either individually or in a combination regimen. Slide number 17 then briefly summarizes what we learned about the vaccine classes. There are, of course, numerous vaccine classes and product attributes, and many of them appear in a combination. So you can have vaccines which are split and highly purified that need to be adjuvanted as well. You can have a recombinant vaccine, which is a VLP, which is also adjuvanted. There may be whole virion vaccines which are adsorbed. There may be recombinant antigens which are adjuvanted and adsorbed, and so on and so on. So it's very interesting to read the summary of product characteristics of certain vaccines in order to learn how they are formulated and to find out to which specific class these vaccines actually belong to. Let's then move to slide number 18, which deals with the so-called pharmaceutical forms. That means to discover how vaccines are formulated. So the simplest form is a solution for injection. That means that a given vaccine antigen can simply be dissolved in water or in a buffer solution and comes then as a clear solution. Other antigens are in a suspension. That means it's not really a solution, but it's a more turbid formulation, and others come as an emulsion, specifically those with complex adjuvanting systems where you have an equal distribution of the antigen and the adjuvanting system, but no clear solution anymore. And that's the case for most inactivated vaccines. There are even more complex pharmaceutical forms, and one nice example is the suspension and emulsion for injection. It's very hard to find out what that means, but it's very easy once you know. So the suspension that relates to the vaccine antigen, and the emulsion is actually one of the novel oil and water-based adjuvanting systems, and that comes in two different vials, two different containers, which need to be combined, or the content of both needs to be combined just before administration. And that results then in an emulsion for injection when you combine both and prepare for vaccination. And Pandemrix, one of the pandemic influenza vaccine used during the recent pandemic, is one of these examples where this complex pharmaceutical form has been used for certain reasons. And the reason is that both the adjuvanting system and the antigen don't stay in this emulsion forever when they are pre-formulated. So for that reason, this concept to have two containers was the most ideal for. Many live attenuated vaccines come as a lyophilized powder, and this powder needs to be resolved by using a solvent like water for injection or buffer systems. That means also in that case, the vaccine is made ready just before use. Slide number 19 continues with the pharmaceutical forms. So many rotavirus vaccines come as an oral solution, which is either fully liquid from the beginning or again needs to be dissolved by using water for injection or a buffer substance, and then and this is prepared right before use. The cholera vaccine comes with granules that actually stabilize the vaccine when passing the digestive tract of a vaccinated individual. So you have granules that actually are dissolved in a lot of water. So you drink this vaccine actually, and again, this water and the stabilizer helps to the vaccine to survive the quite acidic environment of the stomach. Nasal influenza vaccines, there's one product available. It comes as a trivalent and as a tetravalent vaccine. It's live attenuated and it's sprayed into the nose, exactly the entry point where the circulating influenza vaccine viruses normally enter the human body. So it's a local immune response which is set there, which is a very efficient concept, but which is only um, doable for influenza vaccines and not necessarily for antigens used for protection against other circulating pathogens. So slide number 20 then deals a bit with the container and the presentations, how vaccines are actually filled into, and there are first of all ampules which are rarely used anymore nowadays, probably just for water for injection, but they are no longer used because they break easily and during transport and storage, and they have been replaced by vials, which are shown in the lower part of this slide. So many liquid formulations, lyophilized vaccine formulations, and specifically multiple dose formulations come in vials of different size. They have a plastic cap where a needle can be sticked in and the content can be withdrawn several times, though this is much more convenient compared to the previously used ampules. Slide number 21 shows a rich selection of pre-filled syringes. They are all made by glass. They come with a fixed needle, they come without a needle, so the doctor can choose, depending on who is going to be vaccinated, which needle length or thickness is needed. So many liquid formulations, suspensions, and emulsions come in pre-filled syringes. Then live attenuated vaccines. In this case, we already talked about that. The lyophilized powder is normally in a vial, and there may be a pre-filled syringe which is filled with diluent, which comes together with the vial. So that is then used in order to dissolve the lyophilized live attenuated vaccine just right before use. It's very convenient. And finally, slide 22 again alludes to the oral vaccines like rotavirus vaccines, where just a simple pre-filled squeezable tube is used in order to just drop the content into the mouth of a vaccinated individual. That's very easy. And again, cholera, that comes as a bottle, a lot of liquid, and a sachet with a substance that, as said already, helps the vaccine formulation to safely pass the gastric part of the human body. Then slide 23 summarizes the methods of administration. As said repeatedly, intramuscular or subcutaneous injection is the most common form of administering a vaccine. In contrast to the intradermal injection, where only a single influenza vaccine exists, where this intradermal route is actually used mainly for elderly, because there was a slight advantage discovered compared to the intramuscular and subcutaneous injection as regards immunogenicity and efficacy. Nasal application has been discussed also, as well as the oral application, and these are the easiest routes because it's painless, you don't need a specific equipment, and it's just safe, and the patient and the doctor are both very happy about these forms of administration. So, in summary, we can conclude that vaccines are very complex biological medicinal products. As we learned, the drug substance, meaning the vaccine antigen, is difficult to produce and to characterize, like it is for all biological medicinal products. The drug product, which is the final vaccine formulation, which is then administered to a vaccinee, determines the risk benefit ratio of a given vaccine. So the formulation um, defines the risk benefit ratio in a way that it largely um, characterizes immunogenicity as well as safety issues. And this is the most important thing to consider. Again, alluding to the fact that vaccines are given to a healthy population. Modern adjuvanting systems make existing vaccines better and also allow the development of novel vaccines. And this is something where a lot of money is invested in the future in order to address vaccine concepts where no solution has been found yet because antigen production itself is not sufficient in order to make the vaccine effective. And since it is so complicated, there are only very few manufacturers around the globe who are able to cope with the production and procurement of vaccines, um, as well as with the development of new vaccines. So, finally, I hope that has been informative for you. Questions can be asked all the time, and I would like to thank you for your patience and your interest. Thank you very much.